Greetings, and welcome to the lecture on plant molecular biology, focusing on plant tissue culture. In this session, we will explore the fundamental aspects of plant tissue culture, including aseptic techniques, crucial for preventing contamination, and the principles of media design that support optimal plant growth and development. Let's dive in to understand these core concepts that form the foundation of successful plant tissue culture practices. Plant tissue culture broadly refers to the in vitro cultivation of various plant parts, under aseptic conditions, on a nutrient culture medium with a known composition. The excised piece of tissue or organ obtained from a plant for culturing is called an explant. Explants can be taken from many different parts, including portions of leaves, stems, roots, cotyledons, embryos, and various types of mature cells. However, the selection of an explant largely depends on the purpose of the tissue culture, the suitability of the explant for initiating cultures, and its availability. While many different explants can be used for tissue culture, axillary buds and meristems are most commonly used, especially for micropropagation, because they are less prone to genetic changes compared to disorganized tissue. Other desirable properties of explants include being easily sterilizable, juvenile, and responsive to culture conditions. The success of plant tissue culture depends on the choice of a suitable growth medium. Tissue culture media generally contain energy sources or macronutrients, and inorganic salts or micronutrients, to support plant cell growth. The media can be in liquid or semi-solid form, depending on the type of culture. Other requirements for plant tissue culture include growth regulators, biofertilizers, and vitamins. External factors such as light, temperature, pH, and subculturing frequency also play crucial roles in successful tissue culture by supporting plant growth and tissue development. Aseptic technique is essential for the successful establishment and maintenance of plant cell, tissue, and organ cultures. The in vitro environment, in which the plant material is grown, is also ideal for the proliferation of microorganisms. In most cases, these microorganisms outgrow the plant tissues, leading to their death. Contamination can also spread from one culture to another. The purpose of sterilization and aseptic technique is to eliminate contaminants, or minimize the possibility that microorganisms enter, or remain in the cultures. The list of organisms, identified as contaminants in plant tissue cultures, includes bacteria, fungi, viruses, yeasts, mites, and thrips. Contamination with bacteria is considered the most serious, and has been extensively described in the literature. Bacteria are usually introduced with the explant, and may survive surface sterilization, if they are embedded in the interior tissues. Some bacterial spores can also survive the sterilization procedure even when present on the tissue surface. Various types of bacteria have been found in plant tissue cultures, and they can be recognized by a characteristic ooze, in different colors, including white, cream, pink, and yellow. Fungi may enter cultures on explants as plant pathogens. They are frequently present in the soil, or their spores may be airborne. Fungi can be recognized by their fuzzy appearance, and occur in a variety of colors. The next type of contaminant is viruses. Viruses are extremely small, and not easily detected. Therefore, plant cultures are not necessarily pathogen-free even if microorganisms are not visible, and viral contamination can significantly influence culture success. Contamination in tissue culture can originate from two sources, either through the carryover of microorganisms, on the surface or in the tissues of explants, or through faulty procedures, in the laboratory and tissue culture techniques. Most contamination is introduced with the explant due to inadequate sterilization, or the use of very dirty material harvested from the field. Initial contamination is usually evident, within a few days after cultures are initiated. Latent contamination, typically caused by bacteria, may only be observed long after cultures are established. Bacteria may be present endogenously, in the initial plant material, but are not obviously pathogenic in situ. Once in vitro, however, they can increase in number, and overrun the cultures. Latent contamination is particularly dangerous, because it can easily spread among cultures. Contamination can also result from poor sterile technique, or dirty laboratory conditions. This type of contamination is largely preventable with proper care. Whether it is labware, culture medium, plant material, or instruments used in the laboratory, all are sources of infection and need to be sterilized. The routine sterilization procedure for non-living articles, such as nutrient media, glassware, distilled water, and instruments, is autoclaving under steam at a pressure of 15 psi and a temperature of 121 degrees Celsius for 15 minutes. It is important to note that, when autoclaving screw-capped bottles, the caps should not be closed too tightly. This allows gases to expand without the risk of explosion. An alternative method for sterilizing glassware and instruments is heating them in an oven at 150 degrees Celsius for 3 to 4 hours. Thermolabel compounds are often sterilized at room temperature or in the cold by passing them through a bacterial filter. Plastic containers, such as petri dishes or plastic centrifuge tubes, which cannot be heated, are usually sterilized with ethylene oxide gas before being sold and cannot be re-sterilized. Items in the transfer hood or laminar flow cabinet may be sterilized using germicidal lamps. UV lamps should not be used when people are present, as the light is damaging to eyes and skin. Additionally, plants left under UV lamps will die. It is also possible to sterilize items in a microwave when necessary. Plant material that is to be cultured should be surface sterilized to remove surface-borne microorganisms. This procedure should be conducted in front of a laminar air flow or inside the inoculation chamber before the plant material is placed onto the culture medium. Commonly used disinfectants for plant tissue culture include calcium hypochlorite, sodium hypochlorite, hydrogen peroxide, and others. The use of a two-step sterilization procedure, which involves placing the explants in a 70% ethanol solution prior to treatment with another disinfectant solution, has proven beneficial for explant sterilization. For example, commercial bleach contains about 5% sodium hypochlorite, so a concentration of 10 to 20% bleach is equivalent to 0.5 to 1% sodium hypochlorite. Adding a wetting agent, such as Tween 20 or Tween 80, to the disinfectants can reduce surface tension and improve surface contact between the plant tissues and the disinfectants. Conducting the sterilization process under vacuum can remove air bubbles and provide a more efficient sterilization process. The procedures for aseptic technique during plant tissue culture include the following. Turn on the UV lamps in the inoculation chamber at least 1 hour before starting work. Remember to turn off the UV lamps before entering or working in the chamber, as exposure to UV light builds up a high concentration of ozone gas, which is toxic inside the closed chamber. It is advisable to enter the chamber only 15 to 30 minutes after switching off the UV lamps. Power up the laminar air flow for at least 15 minutes before starting work. Place all sterilized articles, such as media, instruments, and glassware, on the glass racks of the inoculation chamber or the work surface of the airflow cabinet. The airflow will then blow bacteria-free air over the working surface. Swab the working glass tabletop of the inoculation chamber or the work surface of the laminar airflow cabinet with alcohol before starting work. Pour alcohol into a clean coupling jar and dip all instruments into it. Flame the instruments using a Bunsen burner or spirit lamp each time before use. Always wear a clean lab coat and mask. Clean your hands with alcohol and allow them to dry. Keep hands moistened with alcohol away from the Bunsen burner. Place the surface sterilized or aseptic plant material in a sterile petri dish. Flame the neck of culture tubes or flasks and quickly remove the plugs from glass vials before transferring the tissue onto the medium, then replace the closures. Lastly, do not dip hot instruments in alcohol, and do not use hot instruments for cutting or holding the plant material. Nutrient medium for plant tissue culture is a mixture of substances on or in which plant cells, tissues, or organs can grow and survive. These substances include a salt mixture, which can be classified into macronutrients and micronutrients according to their relative concentrations in plant tissue, organic substances such as sugars, vitamins, and amino acids, natural complexes comprising growth-promoting or inhibitory compounds, inert supportive materials, and, most of the time, plant growth regulators, to control different physiological responses in vitro and direct the morphogenesis of plant tissues in culture. Plant tissue culture media provide water, minerals, vitamins, growth regulators or hormones, and other essential nutrients, including various organic compounds, for the growth and development of plant cells and tissues. The media also allow access to the atmosphere for gaseous exchange, and act as a reservoir for secondary metabolite waste. The inorganic nutrients in culture media consist of a wide range of mineral salts. Nutrients needed in large quantities, more than 0.5 mol per liter, are called macronutrients, while those needed in very small amounts, less than 0.5 mol per liter, are called micronutrients. Different types of media, their compositions, and the roles of their prominent elements can be found in the literature, and the success of plant tissue culture depends on the choice of a suitable nutrient medium. Basic media frequently used for plant cell and tissue culture include MS, Murishage and Skoog, medium, and Gamborg's B5 medium. Other formulated media, such as NN medium, SH medium, and WPM medium, are also available for different culture systems. Different plant systems have varying nutritional requirements, therefore, there is no single medium that can be used for tissue culture across all plant species. The 6 essential macronutrients for tissue culture are nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. They are required in relatively large quantities, and play important roles in plant growth and development. Their functions range from serving as structural units, to acting as redox-sensitive agents. Generally, the application of macronutrients increases the yield, growth, and quality of plants. Nitrogen influences the rate of plant growth, and is present in the highest percentage of all the essential elements. It is a crucial component of nucleic acids, proteins, chlorophyll, amino acids, and some plant hormones. Nitrogen deficiency is typically characterized by chlorotic leaves and stunted growth, while excess nitrogen promotes vigorous growth, but can suppress flower and fruit development. Common sources of nitrogen for tissue culture media are ammonium and nitrate compounds. Phosphorus is abundant in meristematic and other fast-growing tissues. As part of DNA and ATP, adenosine triphosphate, molecules, phosphorus is essential for photosynthesis and respiration, and it affects plant maturation and root growth. Symptoms of phosphorus deficiency include stunted growth and abnormal discoloration. Potassium phosphate and sodium phosphate are routinely included in plant tissue culture media as sources of phosphorus. Potassium is necessary for normal cell division and promotes meristematic growth. It plays a role in many plant processes, including synthesizing carbohydrates and proteins, and manufacturing chlorophyll. Insufficient potassium results in weak and abnormal plants, sometimes with necrotic margins on older leaves. Common sources of potassium in culture media include potassium nitrate and potassium phosphate, with potassium chloride also occasionally used. Calcium is an integral component of plant cell walls, where it plays a role in the formation of pectin, a substance that bonds cell walls together. It assists in growth, and promotes root and leaf development. Additionally, calcium helps control permeability, and facilitates the movement of carbohydrates and amino acids throughout the plant. As calcium oxalate, it binds oxalic acid, a toxic byproduct of protein metabolism. Distorted or dead shoot, or root tips, can indicate a lack of calcium. Calcium is usually included in tissue culture media as calcium chloride, or calcium nitrate, and sometimes calcium phosphate is also used. Magnesium is the central element in chlorophyll molecules, and is also important as an enzyme activator. Magnesium deficiency causes pale and sickly foliage. Most tissue culture formulas use magnesium sulfate, commonly known as Epsom salts, as a source of magnesium. Sulfur is present in some amino acids and enzymes. It promotes deep green foliage and root development. A deficiency in sulfur results in overall yellowing of the plant. Sulfur is supplied in tissue culture media, in conjunction with other elements, as various sulfate compounds. In addition to the major elements that plants require, several other elements are essential for good growth, but are needed only in extremely small quantities. These are called minor elements, trace elements, or micronutrients. Some micronutrients can be toxic to plants in excess amounts. A few important micronutrients are discussed below. Iron is involved in chlorophyll synthesis, and participates in energy conversion, in photosynthesis and respiration, as it is reduced from the ferric, trivalent, to the ferrous, divalent, state. In tissue culture media, ferrous sulfate is often mixed with the sodium salt of ethylene diamine tetraacetic acid to sequester the iron, making it more readily available to the plants. Boron is an important micronutrient that is believed to play a role in the movement of sugars, water, and hormones. It is also involved in nitrogen metabolism, fruiting, and cell division. A lack of boron results in deficiency symptoms such as tip dieback and chlorotic leaves. However, excessive amounts of boron can cause plant injury or death. Boron is added to tissue culture media in small amounts as boric acid. Cobalt plays a critical role in the overall growth process of plants. It is necessary for stem growth, elongation of coleoptils, and expansion of leaf discs. Cobalt is essential for a plant to reach maturity and for healthy bud development. Other micronutrients include copper, manganese, molybdenum, and zinc. Copper primarily functions as a cofactor for a variety of oxidative enzymes. In culture media, copper is provided in the form of copper sulfate. Higher concentrations of copper may be toxic. Manganese acts as a cofactor for several enzyme reactions, particularly those involved in photosynthesis, respiration, and nitrogen assimilation. Manganese is also important for pollen germination, pollen tube growth, and root cell elongation. It is typically added to the medium as manganese sulfate. Molybdenum serves as a cofactor in two enzymes, nitrate reductase and nitrite reductase, that convert nitrate into nitrite, a toxic form of nitrogen, and then into ammonia, which is used to synthesize amino acids within the plant. It is supplied as sodium molybdate in media. Zinc acts as an activator of enzymes involved in the synthesis of certain proteins, and is essential for the formation of chlorophyll, some carbohydrates, and the conversion of starches to sugars. Zinc is also crucial for the formation of auxins, which regulate growth and stem elongation. Symptoms of zinc deficiency include shortened internodes and smaller leaves. Zinc sulfate is the most common form used in culture media. Organic substances are compounds containing carbon, such as carbohydrates, proteins, hormones, and enzymes. They are not usually provided to plants, because plants typically produce their own food. However, plants in culture are often too small or incomplete to synthesize all the organic chemicals they need, so these substances must be added to the tissue culture media to supplement the plantlet's atmospheric or self-generated supply. Sucrose is a crucial component of nutrient media as an energy source, since most plant cultures cannot photosynthesize effectively in tissue culture vessels. Sucrose consists of two chemically bound monosaccharides, fructose and glucose, and is an indirect product of photosynthesis. A high concentration of sucrose, generally 30 g per liter, 3%, is recommended for most media formulas. Occasionally, fructose or glucose may also be substituted for sucrose in media. Vitamins are organic substances, required for metabolic processes as cofactors, or parts of enzymes. Therefore, for optimum growth, the medium should be supplemented with vitamins. Adenine, vitamin B4 or 6-aminopurine, is important to cells, as a component of nuclear substances, DNA and RNA. It is used in culture media as adenine sulfate to promote shoot formation. Melinositol, a sugar alcohol in the B complex, is required in many media. In its phosphate form, it is part of various membranes, particularly those of organelles such as chloroplasts. Although not essential for growth, it is often beneficial in stimulating cell growth when added to tissue culture media. Thiamine, vitamin B1, is essential for most culture media because it functions as a coenzyme to assist in the organic acid cycle of respiration, also known as the citric acid cycle or Krebs cycle. Amino acids are the building blocks of proteins, some of which combine with nucleic acids to form nucleoproteins. While they are not essential, adding amino acids can augment the nitrogen supply to the media, which is important for specific applications, such as stimulating cell growth in protoplast cultures, and inducing and maintaining somatic embryogenesis. This reduced organic nitrogen is more readily taken up by plants than inorganic nitrogen. L-cysteine, L-glycine, L-glutamine, and L-asparagine are commonly used amino acids, added to the culture medium in mixtures, as they may inhibit cell growth when used individually. A natural complex can be used when a defined medium fails to support a particular growth response, and many such additions serve various purposes. Natural complexes added to the medium generally make it undefined, as variations in growth-promoting or inhibitory compounds in these complexes are to be expected. Some examples of these natural complexes include coconut endosperm, yeast extract, malt extract, tomato juice, banana, potato extract, casein hydrolysate, and fish emulsion. Antioxidants are sometimes used if there is excessive browning of the explant, as they retard oxidation. Examples of antioxidants include citric acid, ascorbic acid, pyrogalol, and fluoroglycinol. Adsorbents are also used to promote morphogenesis, by absorbing inhibitory compounds in the culture medium and substantially decreasing toxic metabolites, phenolic exudation, and brown exudate accumulation. Two common adsorbents are PVP, polyvinyl pyrrolidone, and activated charcoal. Activated charcoal releases substances naturally present in the charcoal that may promote growth. It also adsorbs vitamins and plant growth regulators from the medium and may gradually release them to the explant. Many tissue culture experiments are conducted using various types of stationary supports, such as filter paper, cotton, cheesecloth, vermiculite, and special membrane rafts, with a liquid medium. However, gelling agents are the most commonly used supportive materials. The type of gelling agent used can influence the growth response in the culture. If the gelling agent is unwashed or not purified, it may discolor the medium due to various impurities. Sometimes, dramatic differences in explant response can be observed by changing the brand of the solidifying agent used. Three main types of gelling agents are agar, gelrite, and gel-on gum. Agar is a mixture of polysaccharides derived from extracts of several species of red algae or seaweed. It can easily bind with water, allowing explants to be slightly embedded in the culture medium, while enabling nutrients to diffuse through the medium to the plantlets. Gelrite and gel-on gum are transparent polysaccharide complexes, produced as fermentation products from a Pseudomonas species. These gelling agents form clear gels, unlike agar, which is translucent, making it easier to detect contamination that might develop during culture growth. Although their prices are twice that of purified agar, only about half as much gelrite, or gel-on gum, is needed to achieve the same gel strength. Plant growth regulators are phytohormones used to alter the growth of a plant, or plant part. Many of these are endogenous hormones that are produced naturally in plants, but plants growing in vitro usually do not manufacture sufficient quantities of growth regulators. Therefore, they must be added selectively to culture media. Auxins, cytokinins, gibberellins, abscisic acid, and ethylene are the substances used as hormones, or growth regulators. Among these, auxins and cytokinins are the most frequently used in tissue culture media. In general, auxins promote cell enlargement and root initiation, while cytokinins promote cell division and shoot initiation. An auxin is required by most plant cells for division and root initiation. Auxins commonly used in plant tissue culture media include IAA, indole 3-acetic acid, IBA, indole 3-butyric acid, NA, naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic acid. The auxin 2,4-D is widely used for callus induction, while IAA, IBA, and NAA are used for root induction. However, at high concentrations, auxins can suppress morphogenesis. Auxins differ in their physiological activity, as well as in the extent to which they are bound to cells, metabolized, or move through tissues. Cytokinins are derivatives of adenine that promote cell division and stimulate the initiation and growth of shoots in vitro. Frequently used cytokinins include BAP, 6-benzylaminopurine, 2-IP, 2-isopentenyladenine, kinetin, and zetin. They modify apical dominance by promoting axillary shoot formation. High concentrations of cytokinins inhibit root formation and promote adventitious shoot formation. The ratio of auxin to cytokinin plays an important role in morphogenesis. A high ratio leads to embryogenesis and root initiation, whereas a low ratio leads to axillary bud and shoot proliferation. An intermediate ratio is required for the formation of callus. For morphogenesis, many plant tissues have an absolute requirement for a specific cytokinin, while some tissues are considered cytokinin independent. Gibberellins are plant hormones that regulate various developmental processes, including stem elongation, germination, dormancy, flowering, flower development, and leaf and fruit senescence. They are mainly used to induce plantlet formation from adventive embryos formed in culture. Abscisic acid is used in plant tissue culture to promote distinct developmental pathways, such as somatic embryogenesis, and to culture woody species. It inhibits cell division, and has a favorable effect on abscission. Ethylene is associated with controlling fruit ripening in climacteric fruits, and its use in plant tissue culture is not widespread. Some plant cell cultures produce ethylene, which, if it accumulates sufficiently, can inhibit the growth and development of the culture. In conclusion, the preparation of culture media and the underlying culture technique are inextricably related and impact each other. Even if the culture technique is perfect, the quality of the culture media used can still affect plant growth and development if it is not properly prepared, such as if it is not sterilized, or if errors occur during the preparation process. Conversely, if the culture technique is not properly executed, the consistency of plant tissue culture would be affected. This concludes the presentation. It is hoped that the topic has been both informative and engaging. Thank you for your participation and for joining the lecture.